Welcome dear students. In this video, I'll be going to discuss about previous year's questions of CSIR-UGC NET Life Science, and the topic is plasma membrane. So let's start. Now we will discuss about the second questions that comes in 2018 June part C. There is 2 column, you have to match this column. This is also part C question, that means it actually has a marks, 4 marks. Okay. Now there are options like enzymes, lipases, flipases, floppages and scramblages. You have to know these all enzymes functions, then you can match with this column. So in my plasma membrane notes, I have mentioned this and I have collected the information mainly from Bruce Albert and other books also, like flippage, floppage and scramblage. I have not mentioned the fact that what is lipases, because lipases is very simple. Lipase means that can degrade or actually destroy catalyze the hydrolysis of triglycerides, that means lipid. So the A option is actually with 3. Now you can see, if you know this fact only that lipase means lipid destroyer, so catalyze hydrolysis of triglycerides, then you can see that only A is 3 option is only present in 1, so the answer is 1. So you don't have to know the fact about the flippage, floppage and scramblage, but if you read the plasma membrane notes, you must actually understand what is the function of flippage, floppage and scramblage. You can see here in my plasma membrane study materials, I have write down that flippage is a p-type ATPase. If you watch my transport video, you must know what is p-type ATPase. So flippage is a p-type ATPase, use ATP, and it helps in the movement of phospholipids from extracellular leaflets. Extracellular leaflets means the membrane is actually bilayer. You can see here bilayer. There is one layer, there is second layer. This first layer is called extracellular because this is the outside of the cell, and this layer is the cytosolic layer, plasma membrane. So the extracellular leaflet from the cytosolic leaflet. So that means flippage actually helps in the movement of phospholipids from outside to the inside. Okay. Now here different examples are there, like catalysis of ATP dependent translocation of amino phospholipids like phosphatidylethanolamine and phosphatidylserine from the extracellular to the cytosolic leaflet of the plasma membrane. You can see that this actually information actually true, that is extracellular leaflet to cytosolic leaflet and head group specific PS and PE. So B is actually basically 1, and you can see B is 1 here, only 1 options. And I have mentioned that if you know the simple fact that lipases actually catalyze hydrolysis of triglycerides, you can actually attend the questions. This is very simple, and in many times I have seen that in CSIR part C questions, if you know the very basic fact, then you can actually attend the questions. Okay. So yes, so floppage, how floppage works, you can see here, floppage is a ABC transporters. That means it actually another type of family. ABC means ATP binding cassette proteins. Cytosolic, it helps in the movement of phospholipids from cytosolic leaflet to the extracellular leaflet. So it actually basically performs the opposite function of flippage. So floppage is opposite of the flippage. It actually moves the phospholipids from this layer to this layer. So now we will match the floppage also. Catalysis of ATP translocation of plasma membrane phospholipids from the cytosolic to the extracellular leaflet. So C is equals to 2. That is good. That is right. And scramblage. What is scramblage? Scramblage is also a type of enzyme that actually helps in the movement of proteins, phospholipids from either both sides. So scramblage is actually both bilayer. It moves both bilayer, not specific for particular phospholipids also. Yes. So you can see here that scramblage actually catalyzes the movement of any membrane phospholipid across the bilayer down its concentration gradient. So the option 1 is right. So this is the questions from 2018 plasma membrane CSIR-UGC NET Life Science. Thank you for watching this video.